Hello, and welcome to Season 2 of the Canadian Epigenetics, Environment and Health Research Consortium, or CEE-HRC podcast, presented by their trainees. The goal of this podcast is to chat about some of the explosive and infinitely interesting research happening in the realm of epigenetics, and particularly among the brilliant scientists here in Canada. We hope you enjoy this episode.
Hello everyone, this is your host, Wajih Jauhar, a PhD candidate at McGill.
Hey everyone, my name is Gurdeep Singh. I'm co-host for this podcast. I am postdoctoral fellow in Apprecial Lab at BC Cancer.
We're very excited today to interview Dr. Jacques Coté. Dr. Coté, bienvenue and welcome to today's podcast.
I'm really glad to be here and to do this.
Thank you. We're glad to have you. So before we start, could you please introduce yourself?
Sure. So I'm Jacques Coté. I'm a professor at Laval University, and I'm also an investigator at the Laval University Cancer Research Center and the CHU de Quebec Oncology Division. I've been doing research as an independent PI for over 25 years.
To kick this off, we would like to get more acquainted with you. The journey to becoming an established scientist is quite long. So how did this all start for you?
Well, for me, quite early. I was in high school, in a private high school, and the biology class was pretty amazing. We're like studying genetics and development using like fruit fly and worm and insect for segmentation. And early on, we're doing like genetics with fruit fly, looking at the red eye or white eye and all the F1 and F2 degeneration, all the genetic segregate. And then also understanding the segmentation development, where the legs appears versus the wings and stuff like that. So that was pretty cool and got me really excited and decided early on to go in genetics. And also really being interested in basic research, basic concepts, understanding the mechanism of cell lives. And of course, later on, Drosophila fruit fly was a perfect system also to examine epigenetic mechanism, because that's one of the first example of position effect variegation and having there was clonal red and white eye pattern in mouse and in fruit fly. So that was definitely something that pushed me more towards the understanding epigenetics in the cell and in development as well. So that's pretty much how I got interested very early on.
Yeah. Were you interested in anything else, like beside biology during your early days, or something like shaped your career or trajectory towards epigenetics? Like some milestones which made you go more towards
outside science or in other
outside science? Yeah.
Yeah, I was very much into history and geography. In fact, I was even much better in that. But I guess at that time, in terms of career opportunities, it was less obvious in a very sedentary Quebecer French Canadian aspect of things, where people move much less or tendency to stay in locally. But I think the basic science was definitely always up there with that, and that became natural afterward.
Awesome.
As I was saying, it is indeed a very long journey to becoming an established scientist. And during that journey, we go through high school, undergrad, and then postgraduate training. During all of this period, would you say there was an inflection point, or someone who had an impact on you, a previous supervisor, or a certain discovery that you've made that helped shape who you are today?
Oh, definitely. At different stages. Like I said, there was the high school biology class with the teacher there that was amazing. And then, in undergrad, there was one protein biochemistry teacher that was also like really focused on basic research and got us excited about the basic research, which was very important. And then, of course, getting into grad school, I started as an undergrad in a lab that worked on chromatin, on chromosome structure, basic chromatin, histone DNA interaction. And so I started with that. And that supervisor was really very strict about how to do experiment, to do it perfectly with all the controls. It was even like over the board how much control. I mean, it took me 7 years to do my PhD, so that's part of reflecting that. But it was a successful PhD, but definitely that was a big part of training me as a scientist, how I see how experiments should be done and what are the control and trying to be as sure as possible of the conclusion you're going to be drawing from the experiment. And so that was definitely something very, very important. And then going to postdoc afterward, the supervisor, Jerry Workman in Penn State University, was not as strict, but more innovative in his thinking and more, exchange with the trainees about how the project goes and how the science should be done. Less like a direct supervisor telling you what to do compared to somebody who's really working with you to design the science and do the experiment, and very understanding. So I think at the end that placed me a little bit in between. Like I still retain the very strict scientist critical sense from PhD and the more, teamwork, understanding, discussion, exchange of my postdoc and trying to be innovative in the kind of research we're doing. And that's how but always keeping the very big interest in basic research, understanding molecular mechanism and stuff like that.
No, that's great. So how was your transition from like a very good researcher, like during your postdoc and your PhD, to accomplished scientist right now as a professor? What are the advice you can give to the people like me, like who want to go into academia as a faculty member?
Oh, well, that's I mean, every individual is going to be different, and the situation, family situation, and there's so many factors playing a role there. I mean, to be honest, my PhD supervisor was not a very good one in terms of personal relationship. He was more in the old style of like dictator and being rough and throwing people out. And I hope that in 2023 that doesn't exist anymore and should not exist anymore. But though at the end of my PhD, I had big choices to make because say, okay, do I really want to do that? It seems really tough and painful. But what always got me going was like the beauty of the freedom of doing basic research and make hypotheses and test them and do what interests you the most. And that's something that academia can give you. And of course, academia is tough because you have all those steps before getting advancement. And it would be so often easier in the industry, another choice. But I think that was definitely the freedom of doing what I want and the way I want, and to really decide where to go and what to investigate and things like that. That was the key factor to decide to go to academia, even though even today it seems even more frightening than before and that kind of thing. But I think that's definitely a good. I had offer from after a postdoc from doing research for Red Cross and managing a huge biobank from my home region, which is Saguenay, which is a perfect human genetics laboratory because of inbreeding and stuff like that. But you need to define what really gets you going. That's the key there.
That's true. Yeah, identifying that passion is very important. Yeah, no, absolutely.
Liking what you do, and of course, there's always going to be some aspect of what you do that don't like that much, but if the main thing, the core in the middle is what you like the most and get excited about. I hate what I do in my office when I take care of a lot of stuff, but I love when the student come into my office and talk to me about what their result is and where to go from there. And that's the most fun part of that.
Yeah, absolutely. That discussion is always like very fruitful and like fun. So a follow-up question on that, what is the most stressful aspect like of your job?
Well, I would say different things. There's always the typical thing, getting the funding, getting the papers accepted that are linked to the funding at the end. And I've never really stressed too much about, promotion. I think, well, you won't get unemployed with a PhD or things like that, right?
Yeah.
But I think I felt stressed as much about being able to provide the training and get the training in the lab to graduate with the tools they need to do whatever they want. So that's my agreement with the, I'm trying to provide the resources, I try to provide the guidance, and my job is in big part be sure that they're going to be successful and get the training and the CV at the end of their training, that whatever they want to do, the doors are not closed, that they can go academia, they can go industry, they can go teaching, they can go pharmaceutical and stuff like that. I think that to me was always, and the thing that when you're in academia, you're not really trained for human resources. You're not really trained for financial management. So that was tough as well, because finding every single trainee their button to get them motivated and going and having a good relationship is not always easy. But so we continue to be trained as PI. We're getting trained as much as our trainees in those aspects as well.
Yeah, no, that's a beautiful vision, and great advice. Yeah, awesome, thanks.
Just going back to what you've just mentioned, I think two main points got my attention. First, we're very happy to hear that you're an advocate of healthy supervisor-supervisee relationships. I think that's very essential, and it helps pushing research forward. It helps students to thrive and results to flourish. So that's really great to hear. And then the second point, you also mentioned how important it is to be a bit more malleable or flexible or open to different things, because I think that's very essential for basic sciences. If I'm not mistaken, you were one of the very first people to characterize the BAF complex in Saccharomyces. I think that is something that requires a lot of academic rigor, but also some open-mindedness.
Yeah, but one thing I say too, if you bring back that, I graduated in too many years ago in a PhD. Like I said, it took me 7 years. I finished with a big paper, right? But that paper is 6 years of work. And then I started postdoc. I got in Jerry Workman's lab. He just started his lab. I was his second postdoc. I was very well trained in lab work. And I arrived when, okay, let's purify that Swiss-Schnif complex from yeast. And the genetic was already suggesting that it was involved in changing the chromatin structure and regulating gene expression. And pretty much it just dropped on me, that project, because I arrived there at the right time. I had the ends to do the experiment. So, and that's one thing I said to a lot of trainees, you can be really, really gifted, but if you're unlucky, you can still suffer a lot. And as much as you might know, a lot of time things don't work while they're supposed to work. Even the basic stuff. Even here, we got trouble for cloning for 2 months for some reason. And we're in 2023, we should not have any problem cloning. But so there's so many things that can go wrong in the complexity of molecular biology and genomics and stuff like that, that there's a lot of luck and bad luck going on. And when it works your way, you need to go full ahead with it, because often it doesn't work, even though you have everything to make it work. And that's right. Yes, I've got involved in that, and it's considered one of those 3 papers at that time that discover Switch-NIF BAF complex, and I work on the BAF complex in mammal as well. That was a big thing there, but I was there at the right time on the right project. I just have the ends to do the experiment, but I was not the guy who thought about everything and was innovative then. That was more my supervisor, right?
That's a nice way to put it. And it sounds like a good selection of a supervisor to work with.
Yeah, but you were very highly trained also, which made your success.
Absolutely. Okay, so now to continue off with our interview, I would like to ask you a few specific questions about epigenetics. You've explained to us several times now your interest in epigenetics and basic mechanisms. So from your perspective, would like to hear, what is your definition of epigenetics?
Yes. So, you know as well as me that there's been many definitions, and definition has been used in different ways and debated by other group. I'm more the classical kind of epigenetic believer, and I'm more like at the cell identity memory. So the fact that, you know, when I teach epigenetic, the first thing I say, okay, you have your body, different tissue, all the same DNA, but you have different type of cells, and the cells remember what type of cell there is. So there's other mechanisms that are not DNA based that keep the memory of the cell identity. And that's how I see epigenetics in my interest and what I want to understand. The fact that there's that heritable information that's not DNA based, that is other mechanism. And that's mostly what I focus in terms of my definition of epigenetics. Of course, there's all the aspect of differentiation development, but also what I might mention more later is the effect of lifestyle and environment, because of what we now understand about epigenetics and how it works, and how lifestyle and environment can have and metabolism can have an influence on the epigenetic system. Of course, there's another aspect that I find highly interesting early on, that's still intriguing me a lot, is things like parental imprinting. And, you know, the fact that we have 2 copies of all genes, but a few genes, like you only express the gene that you inherited from your father, or the gene that you inherited from your mother. And that got me even more interested, because I got a son that got a parental imprinting syndrome, or the Willie syndrome, after, you know, almost 10 years after I started my lab. But I was already working on epigenetics, and then that happened. So that's really something also very, very, and I don't think that it's completely understood how it works either, and why it's specific to mammalian. And I think that's, but I'm not so much into intergenerational or transgenerational epigenetics. I mean, there's still debates, but there's a lot of progress done recently about understanding that. I'm trying to stay away of that and focus on cell memory and transmission of information between cell division and things like that.
So a follow-up question on that. So in the cancers which you're working right now, have you seen like recent examples of like loss of heterozygosity or like allele-specific DNA methylation, which could tell the cause of the cancer pathogenesis?
No, we're not looking pretty much at that. I mean, if you want to link it to what we're doing more recently, linked to human pathology, it's definitely driven by our work that was just studying normal physiological molecular mechanism in epigenetics, epigenetic factor, and then realizing with all the sequencing, the human genome sequencing with patients and cancer tissue, realizing that there's all those mutation and the translocation that involve the protein, the factors that we're working on. And that's where we just got interested. Okay, what does it do to the mechanism, to the process that we're studying, those mutation and those? And it's more in that angle that we came from, studying normal system, normal mechanism, and then okay, we have all this information about mutation linked to you know, neurodevelopmental syndrome and cancer and okay, so what those mutations do to those normal mechanism in the cell, and what's getting screwed up, and how could that explain the pathology, but also, since we're working on enzyme and protein interaction, how we can maybe target those enzyme and those protein interaction to try to alleviate pathological mechanism.
Sure, sounds great. Yeah, awesome.
I actually find it quite interesting how many times we start with studying normal development, healthy physiology, and then from that we start studying pathology. Yeah. And then maybe even exploiting this for therapy or for other avenues. I find this very interesting.
Yeah, and that's why since we never work on DNA methylation and we only work with an enzyme that work on histone, on chromatin, that's why when we never studied DNA methylation because we came from a different place, and that's what we're pursuing. Not to say that maybe one day I'll look at it because there's so much information now available, but definitely not currently.
Okay, so we did hint a bit about what you currently do in the lab, but would you like to talk more about it?
We started the lab thinking that we could not survive in Quebec City working on the BAF complex because there was too many lab working on it, and we're already shown how it remodel chromatin and stuff like that. And so we decided, maybe because I work in yeast, we identify with David Ellis the acetyltransferase complex from yeast back in 25 years ago, and say, okay, I'll start my lab on a specific acetyltransferase complex that acetylate chromatin, and the only essential one for viability in yeast, which was Cat5 TIP60 in normal. So that's how I started the lab. And so we purified the protein complex because everything that we're working on is pretty much like large multi-protein complex that have enzymatic activity to modify chromatin structure or modify the deposit the histone marks and stuff like that. So that's what we did. And then early on, we started doing the parallel, looking in human cells, mammalian cells. Of course, then in human cells, it's getting a bit more complicated. There's more redundancy and, it's not isogenic like yeast. So there's a lot more variation between different type of cell and things like that. So we did that. But what was important is that we also made the shift in the fact that, yes, we're studying gene regulation initially and how those chromatin, those epigenetic marks, epigenetic factor were regulating gene expression to identifying the same activity being involved for DNA repair, for genome stability, both in DNA repair of double-strand break and both in DNA replication. So the same large multi-protein complex that acts in gene regulation also act in opening chromatin and regulating access to DNA during DNA repair and also play a role in DNA replication. That was a big leap of interest at that stage. And of course, when we move on further in the human system, what was interesting too is that people started identifying how the different epigenetic marks are recognized in the cell as a signal, as a signaling event, and the specific protein domain that recognize acetylation, methylation, phosphorylation, methylation. And that's how with those large protein complex we realized that we had so many of those reader domains in those complex that make the complex able to recognize different epigenetic signature to regulate gene expression in specific contexts at specific loci in the genome. And that was a big part of what we're doing in early 2010 and throughout that decade. And later on, then we got interested in all those mutation and translocation involving all those factors we're studying and characterizing what those mutation and those fusion translocation do to those basic mechanisms in the cell. And that's what we're working still on and will continue working on in the next few years.
So in these endeavors, what kind of techniques do you use in your lab?
That's the thing. I mean, it's funny because I have difficulties recruiting students working in yeast because the lab has been quite a binary. There's the yeast people, there's the human people working in human cell and the yeast cell. And then more recently, it's getting more and more difficult to convince somebody to work in yeast. It's because yeast power beside the simplicity was the ease to manipulate genetically the genome, the yeast to do the biochemistry, the entire genome collection of mutants available publicly and that kind of stuff. But with the development of CRISPR, highly efficient techniques to purify protein from human cells, that made and the sequencing ease of and lower and lower cost of large scale high throughput sequencing kind of take away many of the power of the yeast and make it available in human system. And I think that's a big development in the past few years to be able to do, what we used to do, like even like synthetic lethality screen. I laugh a little bit when people talk about synthetic lethality screen in human cell, because this has existed in lower eukaryotes, but suddenly it's like a thing in human cell. So I think that's very powerful, because those synthetic genetic interactions that you can identify through CRISPR screen and easily by the high throughput sequencing is very, very important, and it's gonna already made a lot of very important discoveries and functional link between unsuspected factors that are an impact on epigenetics and pathology. And I think that's definitely something that we use a lot. And the fact that one thing that's really difficult is all those epigenetic factors that are essential for cell viability are difficult to study, because you cannot like knocking them out. And if you knock down them using RNAi, it takes like 2 days and things like that. Well, now you have all those deground technology to like acute depletion within an hour. And then you're not swimming in secondary effect. You're really looking at the target directly of the factor that you were not able to study because it's essential, but you can do get rid of the protein within half an hour to an hour with those deground, and then look directly at what's going on, what's the direct target of those factor. And I think that's what we're doing a lot right now, using CRISPR to mutate human genes, using CRISPR to put tag to purify native endogenous protein, using CRISPR to put deground on endogenous protein and to better look at function, and then, doing the proteomics, the functional genomics. And of course, we're working with enzyme and chromatin, so we're doing a lot of biochemistry with reconstituted purified system in the test tube, and recreate more understanding of what's happening molecularly in terms of interaction and function in the test tube, always, of course, in parallel with more in vivo to support, and then having a much stronger conclusion in terms of mechanism.
It's great that you're using both human cells and the yeast. So are your experiments mostly like bulk, or are you thinking about using single cell technologies also? I think a single cell will be more useful for the human cells maybe, but yeast has like beautiful advantage where bulk methods will work great on them.
To be honest, I'm a little bit concerned to moving into single cell, but I definitely see the power of it, and the even like years ago when people look at single cell in yeast, they saw the stochastic variation from one cell to another, even in isogenic yeast population. So imagine in human cell population. So it's kind of daunting to me. It kind of scares me. to get all that information. I'm more like, okay, I'm going to take the average, the population aspect, like that's what you mean you call bulk, right? The general pattern that you detect in the population. But I definitely see the important information you can get by single cell, and that's definitely a lot of efforts are pushed that way. But I'm not sure my lab is, I think we'll need a little bit more bioinformatic power to attack that aspect of research.
So you just mentioned that single cell technologies kind of scare you. For me, proteomics, protein purification, that scares me. So I wanted to ask you, because this is very far from my area of expertise or training. What kind of skills does this kind of work require?
I mean, it's definitely trying to understand everything that happens in the tube, understanding the role of every component in the buffer you're using, and the role of the different condition, pH, temperature, the thermodynamics of interaction, the salt concentration, and things like that. I mean, you have to be pretty, into details, because often if you think about cooking, look, that's a bit debate about biochemistry versus cooking. Some people say, oh, it's like cooking. No, it's not like cooking, because you're like, okay, I'm going to put a little bit of salt, I'm going to put a little things like that. And you need to be more precise than that. and reproducible. You cannot change the recipe too much. A mistake that often happens that, okay, so if I skip this part, it still works. And if I don't put that, it still works. And then people change the condition and the protocol little by little. It still works, but it becomes grayer and grayer, and suddenly it stopped working. And how come it stopped working? So you change too many parameters. You need to control the parameter in biochemistry. But I think that can apply to almost all aspects of lab research, right? But, you need to like that. And I see today in 2023 that often, the papers, like they go get a biochemical help for collaboration. Somebody is doing a lot of biochemistry or structural biology to complement a lab that's more focused on in cellular or in animal research. And I think that's probably the best way to go forward, to have those interactions between specialized teams with real expertise in a specific experimental aspect, and put those efforts together to really go around a question in many different ways, not only in the animal, not only in vitro in the test tube, but putting all together, and then you have a much more powerful conclusion or model at the end, because you support it with different approaches.
Yeah, absolutely. Like screening in yeast model, like thousands of them, and then using the top hits and applying them on like a mouse model to identify those targets, which will be useful in the human cells. So I think that's a great way of research. Yeah.
And now you can do it in human cells with CRISPR. If you look at DepMap, those 1,500 CRISPR screen, you can get a lot of information there to give you hints of what to look for and what to study for your preferred or favorite protein. That's pretty good source. And phosphorite with all the PTM identify on the protein you're looking at, and of course BioPortal, TCGA. There's a lot of public data that can be the source of information, guide you in your research project.
Yeah, that's a very good point. I find a lot of trainees don't harness what's already out there. There's a lot of data out that we can just exploit for our research. We don't have to redo it every single time.
Yeah.
What would you say is the greatest leap that we've taken in the past, let's say, 5 years in the field, from an epigenetic standpoint?
Well, what I already mentioned about the CRISPR screening and high throughput sequencing, that definitely helped a lot understanding epigenetic mechanism by identifying pathways and related factors at different steps of pathways, of epigenetic pathways. That's definitely a big thing. That's definitely a big thing to be able to do degron on essential protein that are involved in epigenetics. But of course, one major aspect is a better understanding of the crosstalk between metabolism and epigenetics. All those factors that require natural metabolite in the cell, that their availability is going to be changing depending on the cell status, if it's cancer cell, and the impact of lifestyle, diet on the availability of those things. That's definitely something that has been exploding in the past 5 to a bit more than that years, where, showing that, the abundance of acetyl-CoA versus the uptake of glucose from the outside the cell. If you combine those two together, you're going to have an impact on the epigenome, an impact of gene expression, and then, being able to use that as synthetic lethality to treat cancer cells that are more dependent on glucose than normal cells and things like that. That's definitely something that's definitely a way forward for cancer. And of course, all those structural studies identifying the reader domain. We think about BRD4 that has been all over, and all the BET protein being able to block reading of epigenetic marks linked to cell proliferation. And of course, cryo-EM, because as I mentioned, many of those epigenetic factors are in large multi-protein complex. And now with cryo-EM, we're able to go like to a couple of angstrom resolution just using an electron microscopy and visualize those large entity by themselves, understanding how they work, and also bound to chromatin, bound to DNA assembled into chromatin, like in vivo, and better understanding, why there's no, this factor is not present in that part of the genome because this mark is blocking its action on chromatin and things like that. And of course, as a last point, I think there's a lot more understanding of intergenerational epigenetics in the past 5 years. There's been quite stronger studies. I mean, there's always been studies for intergenerational, maybe less transgenerational epigenetics. But in the past 5 years, has been quite breakthrough supporting, definitely demonstrating intergenerational irritable stuff. And a lot more hints now about transgenerational, like going down to a third generation and stuff like that. But there's still work to do, obviously, for that. That would be my list of the powerful stuff that happened in the past few years.
It's interesting, the amount of crosstalk that you were talking about, the amount of crosstalk between different disciplines that help propel the field.
And I didn't mention single cell.
Oh, yeah.
Of course.
Okay. One last question. So would you say you have a favorite lab technique that you would like to talk about?
I think it goes back to early on in my training. I love to purify stuff. I mean, even now, it's always exciting when somebody starts from cells in culture and purify the protein complex involved in epigenetics and looking at the gel and looking at the pattern of bands at the end on the gel. Because in the proteomic era, not many people do that anymore. They just tag a protein, do an IP, and then mass spec it, and then they're like, look at the hits that interest them in the mass spec database. I really like having a purified complex, stable complex. You increase the salt, it's going to be more stable. So to be able to do like 2 chromatographic step, affinity step, and look at the gel and being able, okay, this band is this protein, this band is this protein, and it's all stoichiometric, it's all together. And that's still my favorite thing to look at, I would say. And we're still doing it.
So going back to basics, that's very nice. So aside from fundamental epigenetics or fundamental epigenetic mechanisms, several recent and ongoing work in your lab has been on different cancer types, such as AML and endometrial stromal sarcomas. So what drew your attention to these specific cancers?
I mentioned that the fact that you have a lot more sequencing data publicly available made us realize early on that there was those mutation and those translocation implicating the factors, the epigenetic factor we're working on. And that's pretty much what initiated our interest. That's pretty much what, and the fact is that, for example, in uterine cancer, it was always multiple different translocation with different subunit of the things we're working on, always merge with different subunit of another protein complex. And those two entities classically are linked to activating gene expression versus repressing gene expression. So merging them together, found in cancer, in uterine cancer, was reminding us of the leukemia, acute myeloid leukemia for MLL. So, that cancer, that 70% of pediatric AML is that fusion of that epigenetic factor MLL to different partner. So there's a recurrent mechanism involved there that's pretty striking. And the fact that we saw that in uterine, that specific sarcoma, there was that recurrence even more than in AML. It was pretty striking. And the advantage also of sarcoma versus AML is that normally sarcomas are more stable genetically. So the translocation itself is considered a driver of oncogenesis. So that make it more simple to study and to target. And that was really striking to us. For the AML stuff, it was a bit more a detour in the sense, okay, there was one recurring fusion that was linking a subunit of TIP60 with that interesting protein, but also because when we studied normal regulation of the OX cluster with MLL and another of our activity, we're looking at HBO1, we found that functional interaction, and then it was also published that if you target our activity, you can block the oncogenic mechanism. So that was that aspect that interested in AML, but it was a different kind of road to get there compared to the uterine cancer that we studied and we're still studying. And I think at this point, we're still pushing because there, as many cancer, there's different grade of cancer. So there's in endometrial stromal sarcoma, there's low grade and high grade. And the interesting thing is that what we studied so far is all linked to low grade, and the high grade, the translocation are related, again, implicating the factors working on, but they're merged to another repressive complex, not the same one as in low grade. So it's going to be interesting to see what is different in those high grade versus the low grade. But also the hypothesis initially was like, okay, so if you fuse an activator versus a repressor, you should repress things that should be activated normally, and you should activate things that should be repressed normally. And what we discover is, in fact, it's mostly activating things that were initially repressed and linked to more developmental markers like the OX cluster, like in AML and stuff like that. So that was a question we had. So is it repressing or activating mostly in the mechanism involved? And after studying a little bit patient sample, we confirmed what we saw in the model system, that it was more activation that was important than repression.
That's very, very interesting. And what I also find interesting is the fact that a big subtype or a major cluster of sarcomas are fusion driven, but now we're further unraveling that although there are initially genetic events, the main mechanism is epigenetically mediated.
Yeah.
Very nice.
Is there any genomic mutational background which prefers the translocation more versus the others?
No, we did test the translocation in different cell lines. Well, lab cell lines, so pretty much screwed up cell line to start with, and we could detect it not differentially in different cell lines. So I don't think that the sensitivity to the breakpoint is different. Maybe the fusion in that specific context gives an advantage without creating genome instability, because that was one question when I gave a talk to the Fragile Nucleosome Network from Sharon Dance. Is that okay? But what you're working on is involved in repair of double-strand breaks and genome stability. So what is the fusion doing to the genome stability? And that was a very good question that I don't have answer. We never tested that. We showed all that thing that works at double-strand break and allo resection and recombination and repair. But what do the fusion do in that situation? I don't know. We never looked at it. So maybe that's one aspect that we need to investigate, or maybe replication, whatever.
Yeah, that's why I was thinking maybe some proteins involved in the homologous recombination are NHEJ, so other pathways one of them might be more inclined towards.
Yeah, because we showed that our activity TIP60 target many repair factors at double-strand break. So having maybe the repressor function of the fusion will be enlightened and block recombination or repair and allow more genome instability, maybe. But that's very, very good suggestion and idea, and I should write that.
So have you looked into the role of metabolites, like if it exacerbates the fusion products, or their work on epigenetic rewiring for cancer?
Yeah, well, I think there's something going on. I mean, we did that in yeast, so where it's much more easy to play with the metabolites, Sure. We're doing a few things in mammals, and in fact, one of the fusion that's part of the complex is JASF1, which is a protein that's been linked to diabetes, insulin resistance, and things like that. So we're trying now to understand how that's related by playing, with the amount of glucose, nutrient in the cell, and how it affects the function of the fusion in that sense. And I think that's going to be interesting. And I think that's definitely very trendy and important to do right now to understand that crosstalk. And there's a lot of effort doing right now about, the enzyme, the metabolic enzyme that regulate the availability of those metabolites and how it affects, going from quiescent cell versus proliferating cell, depending on the availability of those metabolites. And I think that's definitely something that's exploding right now. But we're still a little bit shy into doing those experiments. Yeah.
No, that's very cool. Very interesting area. Awesome.
One interesting point that you've mentioned was the involvement of development in these cancers or altered development. So do you think epigenetically driven cancers of adulthood also stem from altered development to the same extent as pediatric cancers?
You mean in the sense that it is going to target differentiation mechanism, allowing
Exactly. Like hijacking normal developmental trajectories or hijacking developmental mechanisms for oncogenesis.
I definitely think that's the case for a big part of what we're doing with the sarcomas thing. I'm not so sure about the other AML that we studied, which is the fusion between Zmin11 and ABTD1, because our analysis of that fusion was mostly showing MYC overexpression, which is like, classical oncogene. But at the same time, MYC is involved in stemness. So maybe, yeah, I definitely agree that it goes along with a lot of models of understanding how oncogenesis, how you go back to more stemness and expressing genes that are more linked to that state. I agree that it's probably a very common thread. I completely agree.
So I did have a bunch of other questions, but you've answered without me asking. Is there something of interest you think you would like to talk about before we wrap up?
I think that people need to be careful because a lot of people in the general public want to hear about the influence of lifestyle and stress and about the epigenetics and how it affects their offspring and things like that. And there's a lot of big splash, not necessarily very solid findings, but they find their way into public press, public news, and that makes people think, oh my God, I should be careful with that. There's a lot of noise in that sense. And it's difficult for the public to distinguish what's solid and what is proven versus just what is a, proposed dogma or something like that. I would say that you need clarification for that. And I think that's something that needs improvement but like I said, I think there's been breakthrough to better understand the intergenerational aspect and, and how stress can affect your epigenome. I mean, I have a poster in front of me that dates back to 2004, with the twins, identical twins, raised in different environment and having very different epigenomes. That's a classical thing. And that's one main flashy proof of epigenetic variation. But you need to be careful with that. And that it's difficult to explain it to the general public what's true and what they should believe. One thing that I think we should always say to the general public is that any excess is not good in terms of lifestyle and environment. And that's true for both genetics and epigenetics, because at the end, cancer is as much a genetic disease as an epigenetic disease, and the two talk to each other, so the two can influence each other, and that's something important to relate to. That's something that would definitely need to be better explained, and I think that's part of what CEHRC does, trying to explain better to the public what is epigenetics and what to retain of what is known and what is understood. And I think that's a very important role that the consortium is doing. So congratulations to you all.
Well, thank you so much.
Before we let you go, do you have any words of wisdom for the listeners?
I would like to ask one last thing. The breakthrough are made in basic science and basic research. And I think that Canada is getting behind by lack of funding, lack of support, both in terms of research grant or trainee support. And I think that the public need to understand that the real breakthrough discoveries that have direct impact on the developing new therapies and new way to treat disease comes from discoveries in basic research. And that's why we need to, instead of having, prioritize very applied research, we need to support more basic research and.
Thank you so much, Dr. Coté. We were very happy to have you today, and we did learn a lot indeed.
Awesome. Thank you so much.
Well, it was my pleasure. It was really delightful to do this, and I really enjoyed it. Thank you so much.
Thank you for listening to Season 2 of the This is Epigenetics podcast. If you like this podcast episode, you can check out our website and future episodes at thisisepigenetics.ca. We hope that you all have a very good day.